Chris, did you get a confirmation of recording? Does it say recording?
Yep.
Okay, perfect. Okay, well, welcome everybody. Nice to see a good turnout today. Can I get someone on Zoom to let me know if you can hear us okay?
Yes.
Perfect. Thank you very much. Yes, so welcome again today to Amber Johnston's master's thesis defense. So really appreciate you being here live and virtually. So for those of you that don't know me, my name's Tom Locke. I'm an assistant professor here at Michigan State University in the Department of Fisheries and Wildlife and Pathobiology and Diagnostic Investigation. So I've known Amber for quite some time now when she first joined the lab as an undergraduate researcher. So before I introduce Amber, I did want to take a moment and introduce her committee members. So first, Dr. Kim Scribner, who is a professor and molecular ecologist here at Michigan State University. I'm sorry, evolutionary ecologist, sorry, Kim, both. And Dr. Andrew Winters from Wayne State University, who's a microbial ecologist. So both of them are serving on Amber Johnston's graduate committee. So next, I wanted to give a quick roadmap for today for Amber's defense. So this is the public portion of the defense. So Amber will be sharing some of the results from her 3 years of research and discussing some of those findings. And then there will be time for questions. After that, so around 4:30, that will conclude the public portion of the defense. And then there will be a closed door session where Amber and her committee will continue discussions of her thesis. So that's the roadmap. Sound like a plan? Awesome. Okay. So now it's my pleasure to introduce the star of this momentous event. So Amber Johnston. So she first joined the lab here at Michigan State in her sophomore year when she was majoring in animal science. And I've been privileged to know and work with her since. So she was really excelling in the lab. She wanted to undertake a independent study as an undergraduate student and ended up doing a stellar job focusing on some fish pathogenic bacteria that are a big problem here in Michigan in our state fish hatcheries. So from there, it seemed like she got hooked on fisheries and fish health research, and she ended up rejoining the lab. And I say rejoining in sort of a funny way because I think it was like a day after you graduated and walked that you were out in the field sampling for your graduate work. So
12 hours later.
12 hours later. So it was a quick rejoin. So since joining the lab, I really have nothing but great things to say about Amber. She's any of you that know her, she's a very straightforward, direct, independent, and driven person. She goes out of her way to support her friends and lab mates and is really a natural leader. And in fact, I'm quite sure that you'll see her in this capacity quite soon in the near future. She's passionate about the health and future of fisheries and their environments. And she's a very effective communicator. She's confident, courageous, exudes perseverance, right, in doing all this in pandemic conditions, and is great at blazing trails. So I'm sure after saying all that, you won't be surprised to hear that following her attendance and presentation at a conference here this past May, which I should say was supported by the Janice Lee Fensky Fellowship, Amber was offered and subsequently accepted a position with USDA ARS, the Aquatic Animal Health Research Unit down in Auburn, Alabama. So very excited for her and that opportunity. So before I take the rest of your afternoon with my introduction, I'm gonna close by saying that I'm really proud of you, Amber, and the floor is yours. So go ahead and take it away.
All right. Whew, I'm a crier, but we're good. All right, let me get this all situated here. So thank you so much for the introduction, Dr. Locke. And before anything else, I'd again just like to acknowledge the support and guidance that I've gotten from my committee over the last few years. So thank you, Dr. Kim Scribner, Dr. Andrew Winters, and Dr. Locke for really helping to guide and facilitate my growth as a researcher throughout this master's project. So with that, as some of you may have already known if you heard me talk before, I'm here to talk to you all about one of the coolest fish in the Great Lakes Basin, in my opinion, and that is the lake sturgeon. If my space bar permits. Okay, here we go. So while I was just introduced by Dr. Locke, I like to start out most of my presentations by just giving a brief introduction of myself. So I'm from a small town in the thumb of Michigan, where I grew up boating, fishing, and just really nurturing a love and passion for the Great Lakes that, unbeknownst to me at the time, would lead me to this room today with you all. So like Tom said, I got my bachelor's in animal science from Michigan State University, where I joined the Aquatic Animal Health Lab as an undergraduate student in 2017. And while I was here, I just really fell in love with what I was doing, and that all culminated into me being brought on as a grad student, where I've since met some of the best people and have made some very impactful memories. So to kick things off, well, everyone here is likely familiar with what this image shows. I'm just setting the geographic scene for my thesis research, which is the Laurentian Great Lakes Basin. And these freshwater lakes are, in my opinion, one of the best places in the world to study fisheries. So what fish was I working with? Like I said before, my research revolves around the lake sturgeon, which is, in my opinion, the charismatic megafauna of the Great Lakes. And as you can see, it's kind of hard not to smile when you're working with a fish this cool. So furthermore, the lake sturgeon is highly unique compared to other Great Lakes fishes. They're not only the largest fish in the basin, but they're also the longest living, where their lifespan can extend past 100 years. And furthermore, they have a series of unique life history traits that have led to their establishment in the basin for centuries on centuries on centuries. For example, lake sturgeon mature extremely slow compared to other fish in the basin. And they're also very fecund, which means especially females allocate a ton of energy towards the production of multiple gametes. And lastly, they spawn periodically, with some females going 4 to 6 years between spawning events. So unfortunately, lake sturgeon have a tumultuous history in the Great Lakes, and their populations currently aren't doing very well. The story of their declines begins centuries ago, when they were considered a valuable commercial fish to some for their eggs and meat, while simultaneously being considered a nuisance species to those fishing for other fish in the basin, because due to their large size and bony structure, they would often get tangled up in fishing gear and destroyed as a result. So other factors have also likely contributed to their declines, including habitat destruction. Oh, I don't know what I just did. There we go. All right, so what I'm showing you here is a photo of a lake sturgeon in its ideal spawning substrate, but this clean, rocky substrate wasn't as easy to come by in the 18th and 20th centuries when industries were booming all around Michigan and the Great Lakes. Furthermore, the construction of dams has also posed challenges and sometimes isolates lake sturgeon from their ideal or native spawning grounds. So for a combination of those reasons, lake sturgeon populations sharply declined. And here I'm showing you a graph of commercial harvests by thousands of pounds caught from the 1870s to around the mid-1900s, where you can see just how abundant lake sturgeon once were, followed by steep population declines. And while we don't commercially fish for lake sturgeon in the Great Lakes anymore, and we haven't for a very long time, this graph still does a good job showing how abundant they were, followed by those declines. And unfortunately, those same life history traits that had historically sustained lake sturgeon populations in the Great Lakes Basin are now likely contributing factors behind their long-standing population declines and their long road to recovery. So as a result, lake sturgeon populations are estimated to be less than 1% of what they once were, and substantial resources are being allocated to the hatchery rearing of lake sturgeon for a multitude of things to conserve lake sturgeon, including habitat restoration efforts, the conservation of spawning adult populations through fishing regulations, as well as the hatchery rearing of lake sturgeon to replenish existing adult populations. So when I say replenishing adult populations, what do I mean here? So in the Great Lakes, resources are allocated to the propagation of juvenile lake sturgeon in rearing facilities with the intent of releasing them back into the wild. And there are two main ways that this happens. First, wild adult lake sturgeon are temporarily captured, where eggs and sperm are collected and fertilized and raised in rearing facilities. And from there, fish are hatched, raised for a few months until risk of predation is a little bit lower, and released. Additionally, facilities are populated via fish captured in the wild that are naturally spawned in their larval drift phase, where they're captured, brought back to the hatchery, raised, and released back into those adult populations. So while innovative, these methods mean that whatever problems or pathogens exist in the wild adult populations have a one-way ticket right into those rearing facilities if we're not sure what's going on. So while hatchery rearing of lake sturgeon for conservation is a pretty common practice all around the Great Lakes Basin, most facilities, if not all, grapple with occasional sizable mortality events. And unfortunately, using routinely employed laboratory tests, the cause sometimes goes undetermined. So while this is happening, sturgeon aquaculture is increasing rapidly all around the world, including in North America. And with that, we're seeing an increase in learning more about sturgeon pathogenic viruses, or pathogens that are negatively affecting sturgeons. And alarmingly, some of these sturgeon pathogenic viruses appear to be increasing in both host and geographic range. And despite an apparent encroachment towards the Great Lakes, some of the diagnostic tests that we employ here don't always detect these pathogens, so their presence in the Great Lakes Basin remains largely unknown, or remained largely unknown. So with that unknown in mind, I was afforded the opportunity to conduct a large-scale lake sturgeon infectious disease study funded by the United States Fish and Wildlife Service Great Lakes Fish and Wildlife Restoration Act. And this project looked to assess the risk emergent and endemic fish pathogens posed to Great Lakes lake sturgeon populations. So while my thesis is broken up into 5 distinct chapters, today I'll be breaking out of that mold a little bit and will rather just tell you all the story of this disease surveillance study that for one of the very first times addresses the question, are viruses negatively affecting the health and survival of adult and juvenile Great Lakes lake sturgeon? So to begin addressing this question, I developed a series of objectives. First and foremost, I needed to elucidate the presence of sturgeon pathogenic viruses in Great Lakes lake sturgeon. So jumping right into the methods, throughout this study, I sampled adult lake sturgeon in the St. Clair River in 2019 and the Black River in 2019 and 2020, both of which encompassed distinct spawning lake sturgeon populations, as well as 2 Great Lakes watersheds. While some of you may have seen this photo before, I feel like I'd be remiss not to include it because it just about perfectly sums up the joy that was early pandemic fieldwork in a nice May snow shower. So through this, I collected an array of tissues and fluids from 292 Great Lakes lake sturgeon. I non-lethally collected blood, gametes, and lesion tissues, which amounted to about 470 total samples. Right. So I had all of these samples and was primarily interested in searching for viruses. So I turned to one of the gold standards for virus recovery, which is virus isolation. So this process starts with a tissue sample that's then processed. And one thing about viruses is that they need live cells to replicate in order to grow and be propagated. So to facilitate this, virus isolation also requires the growth and propagation of cells, in this case, fish cells, which the processed tissue sample is then inoculated onto. So if there's active virus present in the sample, it will infect the live cells and those cells will start to die or show morphological changes shown here in this photo, and we refer to these as cytopathic effects. Furthermore, if the cells appear unchanged, it could be for a multitude of reasons, but the 2 I'm focusing on here are 1, active virus might not have been contained in that sample, or 2, the virus that was in that sample wasn't able to be propagated on the cell lines that I chose. And obviously the latter is a problem, and given viruses are generally host-specific, I wanted to put myself in the best position possible to recover viruses that affect sturgeons. So for that reason, I not only employed routinely used cell lines such as EPC and CHSC, and I know those acronyms might not mean something to everyone here, but I also employed 3 sturgeon-specific cell lines that had never been used in the Great Lakes Basin. So often when there's evidence of cytopathic effects of a virus on cells, it doesn't tell you which virus it is. It tells you that something's there, but it might not be indicative of exactly what you're dealing with. And also, if you're not positive the virus you're screening for will replicate on the cells you're using, or if the virus you're screening for even grows on cells in the first place, using molecular tests allows you to look for a virus in a way that doesn't require replication on cells. And these often come in the form of polymerase chain reaction assays or PCRs. All right, so what was I looking for here? One of the first pathogens of concern was frog virus 3. And this iridovirus, as the name implies, was first recovered from frogs. However, it since demonstrated a wide host range. And furthermore, it was recently detected in diseased and moribund pallid sturgeon in the Missouri River. So given this host expansion, in addition to an already vast geographic range, the presence of this virus in Great Lakes lake sturgeon was an important unknown. So to address this, we looked for this virus both in vitro and via molecular methods. So in addition to frog virus 3, I also set out to look for nucleocytoplasmic large DNA viruses. And NCLDVs are a group of viruses consisting of multiple families of viruses, where they earn this name given their replication in the host cytoplasm, in addition to their giant viral genomes. And this group of viruses houses some particularly problematic sturgeon pathogens affecting other sturgeons, and alarmingly, one NCLDV detected in Lake Sturgeon just northwest of the Great Lakes Basin was recently reported in Manitoba, Canada. So given this, we set out to employ specialized diagnostic tests that also were aimed at the detection of NCLDVs. So just of note, I first ran all of these molecular tests with corresponding negative and positive controls to make sure all was well. And when I looked for frog virus 3 in vitro in samples I collected from all of these adult lake sturgeon, I saw no evidence of cytopathic effects on cells, or CPE. Furthermore, when I screened for this virus via molecular methods, I also did not detect any evidence of virus. So similarly, when I looked for NCLDVs in vitro, there was also no evidence of cytopathic effects. However, some NCLDVs are amongst those viruses that cannot be isolated on cell cultures. So for that reason, I also employed a molecular test to look for these viruses. And when I did that, they were also undetected in all of the samples that I had screened. So many of these assays I was deploying were most commonly used on lethally collected samples, such as olfactory tissues, visceral organs, or large pieces of the gills. However, given I couldn't lethally sample adult lake sturgeon, and also to gain insight into what could be going on in these streamside breeding facilities, I also collected juvenile lake sturgeon from 4 different breeding facilities across the Great Lakes, encompassing the Superior, Michigan, and Huron watersheds. So with that, I collected, euthanized, and harvested tissues from 600 juvenile lake sturgeon, and the tissues I collected were visceral organs in addition to fin, gill, and operculus samples. So I screened these samples the exact same way, where I started with in vitro virus isolation and followed that up with molecular analyses. When I looked on cells, there was no evidence of cytopathic effects for either of these viruses, and furthermore, when I looked molecularly, I again found no evidence of these viruses. So from this, we concluded that based on these analyses, there was no evidence of frog virus 3 or NCLDVs in the Great Lakes lake sturgeon sampled throughout this study. So now that I've had the chance to cover what I was looking for and didn't find, I'm going to be shifting gears a little bit and moving into something that I wasn't necessarily looking for, but did find. So while sampling adult lake sturgeon, I observed evidence of skin damage in the form of apparent cutaneous thickening. And these lesions varied in intensity, ranging from focal to multifocal to coalescing in nature. And these lesions also varied in prevalence. So in the Black River in 2019, they were slightly more prevalent than they were in the St. Clair River. And in 2020, it had about a 14% prevalence in the Black River. And fish weren't sampled in the St. Clair River in 2020, given the circumstances we are all very aware of at this point. So through reviewing the literature, I noted that these lesions were highly similar to those induced by aspen-centered herpes viruses. But why did we care about these viruses? Well, aspen-centered herpes viruses are a notoriously problematic group of pathogens in sturgeons across North America and have been linked to mortality events in adult and juvenile sturgeon populations. Furthermore, one of these viruses, aspen-centered herpes virus 2, has also been detected in reproductive fluids, raising concerns that it could be transmitted from parents to progeny. So armed with that information, I optimized a PCR assay using a primer set designed by Dr. Larry Hansen and colleagues that targets the DNA polymerase gene of a range of fish viruses, including those in the family Alloherpes viridae, which encompasses aspen-centered herpes viruses. I screened tissues collected from these lesions, and I did successfully detect an amplicon from an unknown virus associated with these skin lesions. I then sequenced the amplicons, analyzed them phylogenetically, and what I'm showing you here now is the resultant dendrogram. So in brief, I compared my sequences of a partial stretch of this gene to other Alloherpes virus genes of other genes like this from other Alloherpes viruses. And I'm showing you here my sequences all the way over to the right, and from this we gathered that the sequences I had found were indeed distinct from other known aspen-centered herpes viruses, though they appeared most closely related to aspen-centered herpes virus 1. So while I found genetic traces of a virus, unfortunately, and despite my best efforts, I was unable to isolate this virus in vitro at the time, which is critically important for further virus studies. And though disappointing, this wasn't necessarily surprising, given that Alloherpes viruses are amongst some of the most notoriously difficult to isolate viruses in the virus world. So despite no virus isolation, this finding was still a pretty big deal, because aspen-centered herpes viruses 1 and 2 formerly known collectively as white sturgeon herpes virus, highlighted here, are currently listed as emergency pathogens on the Great Lakes Fisheries Commission Fish Health Committee Fish Health Model Program. It's a mouthful, but they earned their spot on this list due to their ability to cause mortality in other sturgeon species, as well as, like I mentioned before, the possibility that they could be transmitted from parents to offspring. And at the time of this initial detection, there were no published reports of an aspen-toed herpes virus or anything of the sorts in Great Lakes lake sturgeon. So my next goal became to isolate this virus in vitro. What I had done in 2019 hadn't worked, so when I was in my field off-season researching various ways to improve virus isolation, I developed modified sample handling protocols. So in brief, I continued to use sturgeon-specific cell lines, including one directly derived from lake sturgeon sent to us from colleagues at UC Davis. I also developed a transport media to facilitate virus growth from the point of collection to the point of inoculation onto cells. And I did all of this because herpes viruses are generally considered weak in the realm of viruses, so I essentially was doing everything I can to baby this virus and make sure it was still active when I got it to cells. Along those lines, I also took any means necessary to avoid the freezing of cells, which did indeed include shipping samples back to the lab every 3 days for about 3 weeks straight. So a lot of logistics, but it did indeed pay off. So after implementing modified sample handling protocols, I inoculated lesion tissues onto several cell lines, including those derived from sturgeon specifically. Soon after, I saw evidence of a replicating agent in the form of multinucleated giant cells, indicated here with the blue arrows. Eventually, that became even more widespread, and as you can see here, that CPE worsened. And I also confirmed via PCR and gene sequencing that the virus I had found in cells was indeed that I found in the tissues. And this finding was super exciting because my new sample handling protocols appeared to have worked very well, and this opened the door to so much more research that I can now do with this virus. So to begin understanding the effects of this virus, we went back to tissues collected directly from those skin lesions. I'd seen gross changes in the skin caused by these lesions, but wanted to know what was happening on the microscopic or tissue level, so I looked via histopathology. But before we get to what I saw in the lesions, here I'm showing you a skin section collected from a grossly normal area of skin, where you can see an epidermis of normal thickness. So now what I'm showing you is tissue collected directly from a plaque-like lesion at the very same magnification, where we immediately noticed how thickened the epidermis was, and we also noted inflammation within the epidermis and underlying dermis. Additionally, we found evidence of tissue damage in the form of swollen degenerate epithelial cells, noted here with the arrows. And we also found evidence of intracellular edema or fluid accumulation between the cells, which are these white channels that the arrows are pointing to here. So collectively, what this told us was that in lesions where we'd found the virus molecularly, we also had evidence that there was damage at the tissue level, specifically in the skin, which was a pretty big problem when you think of that in the context of fish, where the skin plays an important role in host osmoregulation as well as pathogen defense. So because of this, we concluded that this virus could possibly be negatively affecting Great Lakes lake sturgeon. So since we saw evidence of a replicating agent on cells and also saw tissue damage via histopathology, we wanted to hunt for this virus visually, and we did this via transmission electron microscopy. So this is basically a form of microscopy that allows us to see physical virions associated with tissues or cultured cells. And when looking closer at the affected cultured cells, we appreciated enveloped icosahedral-shaped virions, which in here appear as hexagons in 2D. So when looking closer at the affected cultured cells, at the skin lesions themselves, we saw the very same virions shown here, again, with the blue arrows. Appear to have lost my mouse. All right, so now that I linked the virus to lesions and recovered the virus in vitro, I needed to do more substantial analyses to truly discern whether or not this virus was novel. So what did I need? Well, looking closer at those who have done other work with characterization and classification of alloherpes viruses, one thing that all these papers had in common was that they centered their genomic analyses around the DNA polymerase and terminase genes. So you could say that these viruses had become somewhat of a gold standard for alloherpes virus classification. Get this back over to my screen. Here we go. So resultantly, we worked with our collaborators to nanopore sequence genomic viral DNA. And in brief, this process works by DNA being ran through a nanopore protein driven by an electric current, where each base pair that goes through is associated with a specific current readout, which can then be plotted and interpreted. And the result of this is a series of reads or strands of genomic DNA, which can then be used to piece together entire genes and genomes. So while we now have all the information to put the entire genome together and are in the process of doing so, we first wanted to focus on these gold standard genes that I mentioned before. So what exactly did those sequences look like? Here's the resultant dendrogram from analyzing the full DNA terminase sequences, and for this analysis, I compared this full gene sequence to the same sequence in other alloherpes viruses downloaded from GenBank. And excitingly, with analysis of the entire terminase gene, the virus that I had recovered, highlighted here, was completely distinct from all other acid-pentoid herpes viruses where there were sequences in GenBank. And this 100 value here basically tells us that these sequences were pitted against each other 1,000 times via bootstrapping methods, and 100% of the time they were distinct from one another. Furthermore, while there were a few full DNA polymerase gene sequences, what we did have was the full DNA polymerase gene sequence downloaded from GenBank from acid-pentoid herpes virus 1, which we believe to be this virus's closest relative. And even as its closest relative, it was still only 81% similar. So with this in mind, and by looking at these two gold standard genes, we concluded that we were in fact dealing with a novel alloherpes virus. All right, so I had found this virus that I detected and recovered from 2 Great Lakes watersheds, and according to all of our tests, it was novel. However, as we all know, the world of research is rapidly evolving, and the analyses I showed you were all fully complete and near ready for publication, when just weeks ago, Fish Health colleagues in Wisconsin published the report of an alloherpes virus recovered from wild lake sturgeon in, like I said, Wisconsin and the Lake Michigan watershed. And in this paper, the authors described the presence of lesions similar to what I noted in this study, and also deployed molecular techniques to screen for this virus. And while they detected genetic evidence of the virus, they did not isolate or grow the virus in vitro, which sort of limited the ability to perform any other experiments or further experiments that required propagation of that virus. So regardless, they published genome information for this virus, which afforded us the opportunity to see how the virus we recovered compared to the virus that they recovered. So we started with phylogenetic analyses that looked at a partial stretch of the DNA polymerase gene. So here I'm highlighting the sequence data that we downloaded from GenBank and was published from the Wisconsin paper. And all the sequences that you see with black squares are sequences that I had generated throughout my study. And when we looked at this dendrogram, these sequences all appeared highly similar. But given they had published whole genome information for their virus, and we had just acquired the same information for our virus, we wanted to look at further genomic analyses using the whole genome. And this is where things got pretty interesting. So like I said, I had access to published genome data for the Wisconsin virus, in addition to the genome data for the virus that I had recovered here. And to compare the two, we determined percent average nucleotide identities, or percent ANIs. So essentially, this analysis compares fragments of both genomes to one another, and the result is a percent value representing how similar these viruses are to one another. So when we compared the entire genome of the virus I recovered to the virus described in the Wisconsin paper, the percent ANI was 84.02%. However, in the world of alloherpes viruses, the criteria for characterizing viruses isn't perfectly clear, so to give us a bit more context onto what this number meant, we also set out to determine the percent ANI between some other viruses in the family Alloherpes viridae. And here I'm showing you one example of those analyses where we determined the percent ANI between channel catfish virus and blue catfish alloherpes virus. And these viruses are both pretty well supported as being distinct viral taxa. And when we looked at the percent ANI, it was substantially higher. So these two viruses that were supported as distinct viral taxa were actually more similar to each other than the virus detected in Wisconsin and the virus that I had recovered in this study. So from this, we believe that these viruses actually comprise 2 distinct viral taxa. And in this paper that I just recently submitted for publication that's under review as I'm here presenting to all of you, we posit that these viruses be referred to as Lake Sturgeon herpes virus 1, the one recovered in Wisconsin, and Lake Sturgeon herpes viruses 2, the one I've isolated and recovered here. So I found this virus, linked it to potential negative effects in adult lake sturgeon, and showed that it was most similar to a virus recently discovered in Wisconsin, but still appeared distinct. Furthermore, I was able to isolate this virus in vitro, which marked the very first time an alloherpes virus had ever been isolated from Great Lakes lake sturgeon. And this also provided us with opportunities to do further research that we wouldn't have had the opportunity to do if we had only detected the virus molecularly. And a critical unknown that I wanted to address was what effect this virus could be having on juvenile Great Lakes lake sturgeon, especially given that some alloherpes viruses can be transmitted from infected adults to offspring. So with that, I developed the second objective of this study, which was to elucidate the virulence of novel lake sturgeon herpes virus 2 in juvenile Great Lakes lake sturgeon. So a pillar for linking a virus to disease causation is the fulfilling of Rivers' postulates, which requires a virus to be recovered from a host showing gross signs of disease, the virus cultivated in vitro and inoculated into a naive host. That naive host must also start showing signs of disease, and I need to re-recover the virus from that naive host showing those signs of disease. So with that, I began a series of small-scale challenges looking to assess the effects of temperature, dose administered, and route of exposure, all with an eye towards developing the ideal full-scale challenge model to begin fulfilling Rivers' postulates. So based on pilot challenges and because it most closely mimicked exposure in the wild, I pursued an immersion bath exposure experiment. However, this meant I need to cultivate a very large volume of virus to immerse multiple groups of fish. So to put this all into perspective, if you recall, in 2019, I couldn't even grow this virus in my first attempt. And while I did succeed in isolating it in 2020, it was on a very small scale. So small that I needed to grow up approximately 1,000 times what I had currently had in order to even think about doing this challenge. So not only that, because like I said before, viruses require live cells to replicate and grow, I simultaneously had to grow up enough cells to sustain this volume of virus. And I'm not sure how many people have experience in cell culture, are familiar with how the cell works, but this required a lot of effort, and the process of growing up all these cells alone took nearly the duration of the entire experiment. Nonetheless, once I had enough cells plus enough virus to infect those cells, I was finally able to harvest virus and begin this experiment. So for the next few slides, I'll have this little roadmap at the bottom aimed at guiding you through the multiple stages of this experiment. And now with quantified virus in hand, I immersed 6 replicate groups of 8 fish each for 60 minutes in this virus suspension and released them back into their tanks. I did the same and mock challenged 6 replicate groups of 8 negative control fish. So less than 2 weeks post-exposure, I began appreciating a few initial signs of disease, including decreased reactivity to feed, general lethargy, and perioral hemorrhaging, shown here. Perfect, you can see the pointer. So disease continued to progress in 19 days post-exposure. This first mortality presented with more widespread hemorrhaging around the eyes and caudal peduncle, but it also presented with this severe ulceration into the musculature that resulted in near complete loss of the caudal fin. So this continued, and I also appreciated evidence of emaciation. What was most consistent across all mortalities was the severe ulceration of the caudal fin and caudal peduncle. And the development of that same type of lesion continued all throughout this challenge, even as weeks went on. And ultimately, this experiment was concluded at 112 days post-exposure, so just under 4 months. So now what I'm showing you here is the cumulative percent mortality on the y-axis as a function of days post-exposure on the x-axis. And I'd first like to highlight that at no point throughout the course of this 4-month study did I see a mortality in my negative control replicates. On the contrary, when I looked at my virus-exposed replicates, cumulative percent mortality reached near 35%, which when considered in the context of hatchery rearing, the possibility of losing almost 35% of your fish is a pretty big deal. So as fish were worked up either during or at the end of this study, I collected a multitude of tissues for re-inoculation onto cells because, as you recall, to fulfill Rivers' postulates, I needed to re-recover this pathogen from these infected hosts. And so it goes, once I inoculated tissues from those caudal fin lesion samples onto cells, I did indeed find evidence of a replicating agent on cells again with identical cytopathic effects as that observed in the virus initially. And further analyses concluded that the virus I had re-isolated was indeed the novel lake sturgeon herpes virus 2. And with that, I did indeed fulfill Rivers' postulates, linking gross signs of disease to the presence of lake sturgeon herpes virus 2. So we linked this alloherpes virus to the development of gross signs of disease in adults, and we also found it had the capacity to kill juvenile lake sturgeon. So from there, I wanted to figure out how we could mitigate the possible negative effects of this virus in breeding facilities. So given the risk posed by lake sturgeon herpes virus 2 and our ability to now grow the virus and conduct in vitro experiments, we wanted to figure out what this finding meant for those rearing lake sturgeon in the Great Lakes Basin. So a pillar for disease prevention and control is the upholding of biosecurity in these facilities. And the use of disinfectants is an important component behind biosecurity that is used in rearing facilities globally, but also in the Great Lakes Basin. And disinfectants are used for a variety of reasons. They can be used to disinfect eggs in an effort to prevent vertical transmission or transmission from parents to offspring. The disinfectant or treatment of post-hatch fish or eggs that are showing signs of disease, as well as the disinfection of surfaces and equipment used in field and hatchery settings. So given only a few of products are approved for use in aquaculture and hatchery settings, I chose 3 approved for use hatchery disinfectants to see what effects the tools we already have at our disposal could be having on lake sturgeon herpes virus 2. So with that, I developed my third objective, which was to assess the susceptibility of lake sturgeon herpes virus 2 to 3 routinely employed hatchery disinfectants. So to conduct this experiment, I started with the lake sturgeon herpes virus 2 isolate and exposed it to 3 different disinfectants at 2 different concentrations. And I chose these concentrations based on how they're used in rearing facilities or previous research that had been done with them. Furthermore, each disinfectant was exposed for 3 time points, including 1, 10, and 30 minutes. And I want to highlight that all of these different treatments, so look at the Ovidine, like high concentration and time point 1, those were all done in replicate to put a little bit more weight behind the results that I generated. Following exposure for the set time points, disinfectant was then deactivated and our disinfectants and virus suspension was inoculated onto cells and quantified via tissue culture infectious dose 50%, which is a mouthful and you don't have to remember it, but it basically is just a standardized method for virus quantification. And with this, I also inoculated these disinfectants themselves onto cells to make sure that there were no adverse effects in the cells from the disinfectants. So what were the results? Well, to determine how efficacious a disinfectant at any given concentration or time point was, I compared the TCID50 result of virus unexposed to disinfectants to virus exposed to disinfectant. So looking at the results from iodine, I saw that just after 1 minute post-exposure, I saw over 99% reduction in this virus. But looking at these other concentrations, as longer as this iodine sat with this virus, I saw that the percent reduction increased upwards to 100%. And these 2 things I have highlighted here are 2 of the ways that Ovidine is used in hatcheries. So at 100 parts per million for 10 minutes, or at 50 parts per million for 30 minutes. And these results suggested that Ovidine was highly effective at these 2 concentration and time points. And while this is encouraging, there are a couple important things to note here. The first is that iodine only surface disinfects the egg. So if this virus was transmitted in the egg, it may not be as effective. And lastly, while these are in vitro studies and they do definitely show promise, they don't necessarily equate to how this would work in a hatchery setting. So further in vivo studies with real fish eggs and real hatchery conditions are warranted. So moving on to hydrogen peroxide, which is commonly used to treat various diseases throughout different life stages. At 500 parts per million for 1 minute, the disinfectant only induced a 58% reduction. Fortunately, though, when employed at higher concentrations for longer periods of time, percent reduction increased to above 90%. And these results suggest that the longer this virus is exposed to this disinfectant, the higher percent reduction it's likely to be. So similar to iodine, in vivo studies are still warranted to see exactly how these results would translate over to a hatchery condition. However, what I discovered herein was still pretty promising. So lastly, we have Virkon Aquatic, which was highly effective even after just 1 minute. So with this, after just 1 minute of exposure to this disinfectant at these 2 concentrations, I saw 100% reduction of viruses, and there was no active virus remaining when I went to quantify it. So like all others, though, this does still require in vivo work, and Virkon is commonly used to disinfect surfaces and other tools. So this is pretty promising. But overall, while Virkon's hatcheries is still needed with all of these disinfectants, my in vitro results show that each of these disinfectants show great promise in the deactivation of lake sturgeon herpes virus 2. All right, so wrapping everything up here, we have a few key conclusions coming out of my thesis research. The first is that through this project, I've had the opportunity to conduct one of the largest and most extensive Great Lakes lake sturgeon disease surveillance studies done to date. And with that, I employed specialized and specific diagnostic techniques to detect a range of fish pathogens, including some viruses that appeared to be encroaching on the Great Lakes basin. Fortunately, though, when I screened for frog virus 3 and NCLDVs, some of those viruses that were believed to be a potential problem, I found no evidence via cell culture or molecular diagnostic techniques. And with this, these results indicate that there is no evidence of these viruses in the St. Clair and Black Rivers fish that I had sampled, in addition to the juvenile lake sturgeon that I had sampled in those 4 rearing facilities. However, given these recent host and geographic range expansions that I detailed back in the very beginning, I would suggest that these viruses remain on the list of potential causes of disease or things to look for should we ever see mortalities in rearing facilities or wild adult populations. So furthermore, through this study, I successfully detected and isolated a novel alloherpes virus from Great Lakes lake sturgeon inhabiting 2 Great Lakes watersheds. And while a similar virus was recently detected molecularly in Wisconsin waters, this study not only suggests that these viruses are representing different taxa, but also marks the very first in vitro isolation of an alloherpes virus from Great Lakes lake sturgeon. And with that, I've now developed successful methods for virus isolation, propagation, and cryopreservation that will prove to be invaluable for future virus research. Furthermore, by conducting a statistically relevant immersion challenge, we fulfilled Rivers' postulates and definitively determined that this virus is indeed capable of inducing mortality in juvenile lake sturgeon. Which begs the question, has this virus been potentially playing a role in those unexplained mortality events that I'd mentioned before? Or perhaps this virus has been playing in the background, maybe contributing to some of those long-standing declines we've seen in adult lake sturgeon populations. And while the experimental conditions that the experimental conditions that I subjected these fish to may not directly equate to a hatchery environment, when you're looking at the possibility of losing a third of your fish under any conditions, it's still something that warrants a lot of attention. Fortunately, though, not all is bad, because my in vitro studies showed that routinely used disinfectants are showing promise and efficacious against this virus under in vitro conditions. And while I know that these in vitro conditions don't directly correlate with in vivo conditions, those studies are further needed, and the research I've conducted herein is setting us up to do those studies, given we can now propagate that virus. Not only that, the research I've conducted here will also serve as the pillars for the further understanding of this novel lake sturgeon herpes virus too. So with that, I would like to acknowledge the Great Lakes the U.S. Fish and Wildlife Service Great Lakes Fish and Wildlife Restoration Act for funding this study, the Janice Lee Fenske Excellence in Fisheries Management Fellowship for not directly funding this study, but for funding my travel to so many meetings and conferences over the last couple months, the Black River Sturgeon Facility crew, because I truly would not have been able to do the field work I did, especially in 2020, without all of their assistance, Michigan DNR, specifically Gary Whalen, Dr. Ed Baker, and the research crew over at the Lake St. Clair Fisheries Research Station for letting me join them on the RV Channel Cat in 2019 to sample fish. I'd also like to thank my study collaborators, Dr. Esteban Soto and Susan Yoon from UC Davis in California. I'd also like to thank Dr. Matt Griffin of Mississippi State, Dr. Jeff Waldwieser, and Dr. Bradley Richardson from the USDA Agricultural Research Service, Doug Larson from Michigan State University, and also the Black River Sturgeon Facility crew, as well as Dr. Maddie Kupel. I also want to thank current and past MSU Aquatic Animal Health Lab members, because I truly would not have been able to do any of what I've done over the last 3 years without you guys. And I told you all I'm a crier, so here we go. But I'd also like to thank Dr. Kim Scribner and Andrew Winters, Dr. Andrew Winters, for serving on my graduate committee. And lastly, I'd like to thank my major advisor, Dr. Thomas Locke, because like you said, I've been here since 2017, and I wouldn't be the researcher or scientist I am now without his guidance. And this was the one I was supposed to cry on, but I started too early. I also just want to quickly acknowledge the community of people I've made here. Y'all are the best, and I could not have done everything that I've done the last 3 years without the support of all you guys. Now I see my mom crying, and I'm crying even more. With that, I will take any questions.
Awesome job, Amber. You persevered once again to be kind. So. now we have time for questions. So I'll try and monitor on here, but please feel free to raise your hand for questions. And you're definitely getting a stream of awesome jobs in the chat. So
Nice.
I'll try and wade through questions. Here we go.
Question.
Oh, sorry.
Congratulations.
Thank
First
you.
of all, that was an amazing talk.
Thank you.
I guess my question is pretty broad. In the last part where you're talking about the different kinds of like disinfectants and the kind of impacts they had, and I don't really understand the difference between the in vitro and in vivo with the hatcheries. I guess I'm sort of curious about what you might recommend based on that for how people use those, or like something you recommend.
Yeah, so that's an awesome question. So I guess just like first part, so like in vitro basically, so that's what I was doing. I was just exposing them under laboratory conditions. So you could think like pristine conditions, like just disinfectant and virus, just cells, nothing more, nothing less. Whereas in hatcheries, you're dealing with a lot of other stuff. You're dealing with the presence of organic materials, which has been shown to pretty substantially affect disinfectant efficacy. So there are also things that you kind of have to follow when using these disinfectants. They're only approved for use at certain concentrations or certain time points. So I guided a lot of what I did in vitro with those known and approved ways of using them. So that would be where I would start with that. I would look at the in vitro results, use that as somewhat of a jumping off point, and then from there, use that concentration and time point that was most effective and approved to also see how that would work under those conditions when you have organics in play and other things like that that could negatively affect disinfectant efficacy. So,
Thank you.
Yeah.
Other questions?
I'll ask a question.
Sure.
So knowing kind of the general populations of lake sturgeon, would you expect this virus to be found elsewhere? And if so, where would you recommend kind of screening to look for that and why?
Yeah, so that's an excellent question. The unique thing about a lot of the lake sturgeon populations in the Great Lakes is that they are pretty distinct and pretty well separated. So to use what was found in Wisconsin as an example, there's little to no overlap between the populations of lake sturgeon that are hanging around that area in Lake Michigan with the populations of lake sturgeon I was sampling in Lake Huron or Lake Erie, specifically Lake Huron, because that population is completely isolated from everywhere else. But we don't have a good idea of how long this virus has been in the Great Lakes, period. So my suggestion for surveillance would be kind of just to do the best you can to check out a lot of those bigger adult populations and just see if it's already been there or not. In terms of, again, I feel like it goes back to how long this virus has been in the basin, which I don't have a good answer to. I don't think anyone does at this point, but my knee-jerk reaction when resources are not limited would be just check whatever you can, whenever you have availability. But I think in terms of priority, you could also prioritize adult populations where gametes are being used to populate breeding facilities. So.
Amber, I'll try and shout. So you have a question in the chat. It's from Dr. LaFrance. He says, "Great job with what you have learned. How would you design a surveillance plan to investigate the role of this virus in potential disease issues at hatcheries?"
Yeah, so that I feel like jumps off of that question real well. So I think what I would first want to do is when you're looking at where you collect those samples, so looking right at the adult populations that you're looking at, that would be where I first prioritize my surveillance efforts, because in order to see what could be going on in the juveniles, when you're dealing with the virus where you don't know if it's vertically transmitted, you want to see if that virus is also hanging out in the adults. But from there, after figuring out what's going on in the adults, I would say just making sure you're screening those juveniles post-hatch. And I know there's a set number that you can look to be like 95% certain that that pathogen isn't in that group of fish, and I would suggest following that.
The other questions, we still have.
Yeah.
Go for it.
Oh, I was sorry, maybe a stupid question.
It's okay.
Or I missed. I was wondering how you quantify a virus when you did that experiment with this happened. And well, did you have like limits of detection because seeing 100%?
Yeah, so like, are you asking how I quantified virus and the disinfectant efficacy? So yeah, the question was how I quantified virus via disinfectant efficacy, those experiments, and if I had limits of detection. So I'll try and give like a brief synopsis of how the assay works, but pretty much what I'm doing is I'm infecting serial dilutions of virus onto, in this case, I did 4 replicate wells. So I'm calculating that TCID 50 value based on where I'm seeing cytopathic effects. So where I start to see less cytopathic effects, I get down into like the higher diluted samples. So like if you're thinking of it in terms of serial dilutions, like 10 to the minus 5th or like 10 to the minus 6th. So I would see what's going on there and then keep looking. So basically just look for all of those cytopathic effects through the wells. So I guess you could say there may be a limit of detection if I wasn't getting anything on my undiluted samples, and a limitation to those like 50% endpoint things like TCID 50s or LD 50s is that if you don't at any point have 50%, over 50% of what you did infected, so I did 4 wells, if all of my wells were only 1, you can't use the LD50 or TCID 50 formula to calculate that. So that could be a potential error, like something you could look out for. But with my work, I can say that I didn't have that happen. Where I had calculated tissue culture infectious dose 50s, I was able to do so because I did have that gap between where half of the wells were infected and more than half. And for Vercon specifically, again, I did not detect virus in any of those wells. But you're right, if I found it in only 1 or 2 in the undiluted sample or 10 to the minus 1, that would be something that I wouldn't necessarily be able to account for or calculate with TCID 50s. So awesome question.
It's a very good question. Maybe Amber, I'll push. I know I'm not supposed to ask too many questions in the open part, but just can you comment on any differences that you're aware of between using tissue culture versus molecular methods, just in general, like sensitivity-wise, limit of detection?
Yeah, of course. So like I said before, virus isolation is somewhat of a gold standard, but one of the issues or inhibiting factors behind virus isolation is that you need a good amount of virus in order to induce effects in some of these cells. So in general, molecular assays that I deployed here tend to be more sensitive or specific, or not specific, generally more sensitive. So if you have like a small amount of virus that you're looking at, it might not grow on cells, but that might not mean that active virus still isn't there. Like I said before in the beginning, there are a lot of reasons why you might not be seeing cytopathic effects on cells. And one of those could be that there just wasn't enough active virus present. So you could pick that up molecularly. But I mean, fortunately, with a lot of the stuff we deal with in the Great Lakes Basin, we are able to detect that stuff pretty readily in vitro. But there is certainly a difference in sensitivity when you're using molecular versus culture-based diagnostics. So. Yeah.
I don't remember you mentioning a sensitivity analysis with serial dilutions or other things for the molecular assays. Did you do that?
So for the molecular assays, no, I did not. For the assays that I used, well, one was a conventional PCR. So I pretty much just used my positive control DNA and diluted those all to a standard concentration before I ran them. And I made sure that that concentration wasn't less than what my extracted DNA positive control was, if I could. I mean, some of the DNA extracts were too low to quantify, which that happens. So I guess there might be a little bit of a hole in the design there. But in conjunction with the in vitro results, I do feel confident saying frog virus 3, for example, wasn't there. But with NCLDVs, there was actually a really good paper put out by Cluthe et al. Where they did very specific work on the virus sensitivity. So that limit of detection is very, very low for those NCLDVs. So I didn't do any of that work necessarily in-house, but there is a lot of work that has been done specifically with that qPCR. So awesome question.
Go ahead.
You started the work at Black Lake in 2019.
Yes.
It was only a few years before that that the researchers there were describing in field notes these plaque-like conditions that have turned out to be this herpes virus. So Black Lake has been isolated from the rest of the Great Lakes since 1907. How did it get there?
So I don't have a great answer for how it could have gone there, but I feel like my go-to would be maybe it just hadn't been inducing the same signs of disease, or maybe those fish weren't stressed out. I mean, one thing about herpes viruses, and you see the same, I hate to like relate it back to humans, it feels kind of gross, but like with cold sores, you get cold sores when you're stressed out, and that's a herpes virus. So it could have been something that changed in terms of environmental conditions that were just inducing more stress in these fish and led to that virus to come out of latency and begin showing signs of disease again. It could have been there for a really long time, but things could have been going a little bit better for the fish. I mean, I don't know that. I'm not sure anyone really knows exactly how that all went, but that would be my hypothesis, is that it has been there the entire time. It's just been latent, and perhaps something changed or something went on that resulted in the development of these lesions. So
Lots of good questions. We still have a few minutes. Do you want to continue grilling Amber in the open section? If not, I'm not seeing any more in the online chat, so one more round of applause. Amber. You're getting lots of virtual applause too. So.
You should copy the transcript of the chat. She does it before ending the Zoom.
Yes, thank you. I won't end this. So we've got it. Okay, well, thank you. So folks are welcome to hang out. Amber had baked a lot of delicious stuff back there, so you can hang out for 5 more minutes, but then we need to get to the closed door session. Thanks again for coming.
should I wave? Thank you, everyone on Zoom. Hang on.
Yeah, thank you. Thanks for coming.
It's not that it's boring, it's that I have no idea what the heck you're talking about. I know you're talking about fish. Even there are a lot of activities.
Yeah, stop recording.
Great job. How do I exit out? There we go.